Hello, my name is Camden Cooper, and I would like to explain why the greatest scientific developments of the past 5 years are those in the fields of gene therapy and gene editing, especially the development of EvoCast, which has the potential to improve upon and complement current gene editing methods by being both reliable and accurate. However, CRISPR technology is another kind that has developed as a uniquely tailored system as opposed to current methods that are available for the public that are non-specific, and it may sooner become available to the public, even though it can require more time, energy, and effort to provide. Overall, the field of gene therapy and gene editing revealed the ability of the natural world to provide healing for people through its own self-preserving processes, as well as opening avenues for research in other disciplines, helping us to better understand the natural world. So let's get started. CRISPR technology, which was used to save the life of a human patient, the first tailored treatment helped save this baby, KJ Muldoon, and CRISPR technology gets its name from a pattern of bacterial DNA sequences known as clusters of regularly interspaced short palindromic repeats, and the bacteria use this system to protect themselves from viruses. This is an image of the process known as CRISPR-Cas9. So what happens is basically specific sequences of viral DNA that the bacteria has been in contact with is converted into is stored in the CRISPR DNA of the virus. The interspaced part comes from spacer sequences that are in between the palindromic repeats, and I believe it is in the CRISPR part of the DNA, which I believe is the palindromic repeats, that the information from the viral DNA is stored, and that information is converted to RNA, then turned into a complex with another RNA, and that directs then the bacteria is able to direct the Cas9 protein or the CRISPR-associated protein 9 to cut out specific viral DNA sequences. And this can be used to cut out mutational sequences in DNA since it has been evolved for people. However, this requires a lot of time, energy, and effort since it's base pair editing. You're dealing with individual mutations generally, and that can be a problem especially when there's a lot of mutations. So EvoCast may be a more comprehensive and effective method. According to Columbia University Irving Medical Center, casts in their natural form unfortunately perform poorly in human cells, and it can be more difficult. It has been a challenge to edit it into EvoCast because it's more of a genetic diversity mechanism rather than a fast-acting protect from the viruses, which made CRISPR probably easier to develop for people. So PACE was developed, or phage-assisted continuous evolution, which enhanced the rate of evolution to produce EvoCast. And it can also be used as well as inside the body in CAR T-cell therapy. So the methods of a new CRISPR method and EvoCast technology have been the greatest scientific advancements in the past 5 years. And in addition to having enormous implications for medical advancement and further research in other fields, it seems that they show that natural processes like genetic diversity creation and protecting from viruses can really help people. The sources I used were Columbia University Irving Medical Center, bioengineer.com, and livesafe.com, bioengineer.org.